Good afternoon, everyone. Welcome to the kickoff for the DART Tdlc seminar series. First, we'd love to thank DART Neuroscience out there in Dartland for funding our seminar series again. We think you're going to be really excited by the talks we have this year. And we have a great kickoff speaker. Marty Serino is back. And he was very nice to agree to kick off our series here. And many of you already know Marty. He's a good friend to our community and helped to actually found, he was one of the founding members of the Tdlc, pre-move to UCL Birkbeck, which we started calling University of California London at some point because so many of our members went that way. It's hard to give Marty an introduction. He knows just about everything. Just ask him and he'll have something interesting to say. His background goes through geography, paleontology, neuroscience, philosophy, and on through theories about language. And so, I mean, many call him the Renaissance man. He can actually draw, too. It's amazing. So without saying any more, I'd like to introduce him to talk to us today. He's going to talk about its maps all the way up and down. That's way better than turtles. Thank you.
Thank you, Andrea. It's very nice to be back in San Diego, where there's lots of light and other very positive things. And so I'd like to take you on a whirlwind tour of maps in the brain. There'll be time for questions at the end. I'll stay under 400 slides. And so it's a play on turtles, turtles all the way down, turtles all the way up, turtles all the way up. So why focus on topological maps? So maps, preserving maps between areas. It all got started with turtles. So I worked on this system in turtles, which is the attention. It's one of the visual attention systems of the turtle. There's a projection, a cholinergic projection that goes up to a map in the superior colliculus of the turtle. And this is a beautiful terminal from up in the superior colliculus. And you can see it's about 1/10 of a millimeter across. It forms a nice high-resolution map in the tectum. There were non-topographic maps, non-topological maps, like there was one in a nearby nucleus. So I'm not saying that there aren't any, but topological maps are the first principal component of organization of the brain. And there's really no place in the brain without maps. And so let's just look at a couple places you might have forgot where there are maps. So here's a projection from caudate or a putamen neuron going to the globus pallidus. It makes 4 maps inside the 2 segments of the globus pallidus. All the neurons make that. And then they continue on to the substantia nigra. If you go down to substantia nigra, you can see that the projections into the reticulata are topographic from 2 nearby points in the caudate there, but also the projections into the dopaminergic part. So if your index finger did something good, why reward the middle finger? You know, why shouldn't there be maps in the reward system just as well as V1? Or look at the cerebellum there, a giant map. We'll see what the cerebellum actually looks like. If you look at the things that project to it, look at the inferior olive. It's basically a little cortex embedded down in the brainstem. If you look at it in a cross-section, you can see it's all folded up because it's trying to preserve these 2-dimensional topological maps of the cerebellar output and input. Even the olfactory system has maps in it. The receptors aren't mapped, but they sort themselves into a map in the olfactory bulb. So we're going to just take a whirlwind tour, emphasis on the whirlwind, of visual maps, a bunch of other different kinds of maps. And then we'll talk a little bit about methods of doing architectonics in vivo. And then we'll talk a little bit about cognition maps, you know, how much overlap are maps actually sort of important for good stuff? And then we'll take a peek at the cerebellum and mirror neurons gone wild. So starting off in the cortex. So the cortex is a sheet. I did a lot of work in owl monkeys, and I finally published these papers after all these years of marching across the giant deserts of visual cortex, where I would, Monday, Tuesday, Wednesday, Thursday ran into auditory cortex. That's what really sort of hammered home to me just how many maps there are.
That was continuous days, right?
It was continuous, yeah. That's 4 continuous days. So here's a section. There's a sagittal section through the through MT. You can see that big blood vessel at the star. There's the big blood vessel, and some of the maps are pretty obvious. Like the V1, V2 border is the most clear map. It was discovered just by macroscopically looking at brains. V2 anterior border, a little harder. The posterior border of MT is pretty clear. But me and several people, Roger Tutel and Jaime Olavarria, devised this method of peeling off the cortex and sectioning it along that dashed green line, and then you can actually see these maps much better. The V1, V2 one, you can see better. You can see MT quite clearly, but if you look at V2, yeah, you can sort of see, if I give you a little after image there, where the anterior border is, but it's not super clear. And then if you look up, there's a series of maps up there. Those are pretty hard to see. If you zoom in, you can see that you require, a really high density of recordings in order to actually expose these maps. And if you look at the myelin, they're not super clear. So here's that same stretch of cortex mapped out. You can see there's a series of 4 or 5 little maps in front of V2 up in sort of the beginnings of parietal cortex in an owl monkey. So some observations of how these maps are set up. These areas can be quite small. So here's one where the name of the area is wider than the area. It got, it's a little leftover area that got several different names applied to it. But in that little area there, MTC, DLA, V4T, there you can see it's actually narrower in its tangential axis than the thickness of the cortex. And if you look at some of the areas in mice, they might be 0.05 square millimeters, containing an entire map of the hemifield in a mouse. So these are tiny little tubes with a map in them, they're like this big, extending through the thickness of the cortex like that. Tiny little things. So some of them are correlated with myelin border, like MT is pretty good, but unfortunately that's still MT. So John Allman many years ago discovered that the periphery of MT drops its myelination for some unknown reason. So it's pretty hard to actually just use myelin. These areas vary a lot between individuals. Here's 2 different individuals at the same scale, and you can see that area is increasing by about a factor of 4 in another animal of exactly the same size. So they're pretty variable. But you might wonder, how are these things, I'm doing these tiny little penetrations, how could that be consistent with all these patchy connections that we know about? So you know that in V1 you've got blobs and you've got stripes in V2, and then there's patches and interpatches in MT, and then there's all different kinds of little patchy things. And no matter where you go in the cortex, if you inject, you get these little patches. It doesn't matter if you're in S1 or M1 or V4 or V2 or prefrontal cortex, there's always little patches. But if you actually look, you tend to forget when you look at these pictures that more than 90% of the connections are in that little 1 millimeter radius circle. So the cortex is overwhelmingly locally connected. And that's about the size of our standard voxel, so we're well positioned to try to pick up this million neuron voxel, well positioned to pick up this structure. So extending this to humans has to deal with this. So this is quite a mess. And so early on, I took a direct approach. I just peeled it off. I'm good with my hands. This took about 12 hours. And I got it sort of flat, reasonably flat. Cortical flattening in the early days. So that's the calcarine sulcus right there. I painted where the gyri were so I could tell where I was. That's why it's painted. My former advisor said, "You've just shown me that you can turn the brain into liver." And then I also lettered. You can see the little red numbers. I could see. Then they gave me one of these. I also got a very sharp graduate student, my first graduate student, Anders. And although we sometimes got lost hiking, Anders would know what that meant. We never got lost unfolding the brain. I knew the brain could be unfolded because I peeled it off myself. So Anders wrote the core of Free Surfer in just a couple years in the early 90s. But once we started applying this to multiple brains, the problem became obvious. We could inflate them, but they're quite different in their local structure. So the actual structure of the sulci vary, but also all the little crinkles vary a lot. And so not being as good of a programmer as Anders, this was my first attempt to turn the brain into an ellipsoid. It wasn't quite so good, but then I eventually got the ellipsoid equation down, and then if you actually take a measure of sulcal depth, you could use that to stretch one brain into alignment with another. And this allows you to do a strange thing, which is very hard to do in 3D, which is, if you look at that area right up there, you're aligning a sulcus in one person with a gyrus in another. It's very hard to do in 3D. We think that's a good thing because it preserves the neighborhood relations. Bruce Fischel rewrote all these programs much more sensibly and more robustly on a sphere. You can see the same thing on a sphere. Makes a lot more sense. And you can apply a coordinate system and then start to average. But one other thing that's really important to keep in mind here, one of the big advantages of using the surface is that you get rid of all these little crinkles. So there's all these secondary crinkles inside the sulci, which is kind of like a second order of hell of trying to line up data across brains. You can see them right there. You can see all those, that's the parieto-occipital sulcus, and you can see all those little medial parieto-occipital sulcus, all those little crinkles. So those things mess up the data between people as well. You can see it right here. This is the standard Free Surfer FS average. Well, this was made by surface averaging folded coordinates, which gets rid of all the crinkles, but it's a shrunken head because you got rid of all that surface area from averaging all those crinkles out. So the right way to do it is to inflate the brains and then average the surface coordinates, and then you get something that has the right. This one is useful if you're doing stuff in 3D, and then at the last stage for a gratuitous surface display, you sample it onto the surface, but that's not moral. What?
So you showed us that an area could be twice as big in one animal than the other. So
Yep.
even if you have a perfect alignment, doesn't mean you have the same areas on top of each other.
No, absolutely not. So we'll come to that. So then we have to actually sort of get some functional data. This original surface thing was designed for MEG, EEG localization, not MRI. But when MRI came online, we quickly piled out to Boston to start doing it. And early on developed a completely surface-based pipeline where you sample to the surface at the beginning and do everything on the surface to avoid all these problems that I just talked about. So these first experiments were done with very large stimuli to see some of these early visual areas that we knew would be there, but we quickly moved on to higher visual areas and actually used a task, because in the old days, who needs a task? We're biologists. So, but I was in the cognitive science department, so we got a task. This is delayed saccade, and you can see that there was a map of remembered saccade locations in parietal cortex in what I called LIP. This enraged people who studied the human brain because they said, that's not LIP, it's way too medial. So how did it get so medial? Well, there's a relatively large default area, so-called default mode network area in the angular gyrus in humans, and it basically just pushes LIP. You can see the same thing is there in a macaque, but it's smaller relatively. And basically just sort of pushes LIP medial. So LIP, which is supposed to be the lateral interparietal sulcus, ends up being right on the midline. That's why the neurologist didn't like it at first. But then we went on to things like windowed video, sort of naturalistic video, to try to activate higher areas. Or this is what I use now a lot. It's there's the there's a flow field and some checkerboards, and there's some contrast in there, and there's your occasional number and an upside down face. And so what we're doing is we're basically mapping attention. We're mapping sort of attentionotopy. If you're not attending, you won't see these maps. And so if you do that, you see a much greater coverage of the cortex. And I'm going to talk about a number of maps here with red, always upper field, and green, lower field. That's the original color scale, the only one that can be used. Now, this is some work done by Rui-Song Wang, looking at multiple different subjects. And you can see there's a lot of variability here. And that sort of disturbs a lot of people in the human literature, that maybe everybody doesn't have the same number of visual areas. But, that's probably the way it is. There's quite a bit of variability, as we would have expected from the animal studies. This is Rui-Song's 24-subject average. Now, you can see that a number of these areas are well enough aligned so that if you average 24 people, average the maps, complex valued maps in 24 people, you can actually see MT, for instance. MT shows up in a 24-subject average. So they're common enough so that you can see that. There's the original LIP. But you can see there's a whole series of other clusters of upper fields with lower fields surrounding them in cingulate cortex and anterior parietal cortex. And as you go forward, you can see it gets awfully close to the central sulcus there. And it turns out that's right at a place where we would expect there to be a multisensory map called VIP, which had been studied in macaques originally, showing receptive fields that map the visual world, but also have lined up receptive fields on the face and the upper body. So that's where it is in a macaque monkey. It's right at the border between visual and somatosensory cortex. So Rui-Song and I made this fine spider to gently air puff the faces of CogSci graduate students. So it's actually quite a pleasant little cool, we never puff the eyes, a pleasant sort of cool thing. And basically we discovered at least a hemi-face representation in the post-central sulcus, quite medial in the post-central sulcus, that we call VIP, or putative human VIP. So there's the air puffs in a dark map at the top. That's the retinotopic map at the bottom. If you look at the overlap, they're amazingly aligned. This was quite a surprise to people who had been studying it in animals, because they just record here and there and don't sort of map the whole map out. But it turned out to be a reliable map that averages across 24 subjects. It turns out it's not really a retinotopic map, it's a face-centered map. So if you have somebody in the right watch a video while looking at the center of that video as the video moves around the face. So basically, you're keeping the retinotopic stimulation constant, but then moving something to different parts of the face. You see exactly the same map as you would see if you did air puffs in the dark or fixate the center and move something around the face. So it's really face-centered. It's translating things back into where they are relative to my face. If you look at the 24-subject average, you can see a hint that there's actually 4 different transitions from lower field, lower face to upper field, upper face. Rui-Song went on to actually make some high-resolution RF coils to use smaller voxels to really pin down what these areas look like. And as you might expect, they're different in different people. Some of them have that sort of 4 cloverleaf kind of structure, but other ones don't. They just don't. Maybe they run their head into the door frame more. My VIP is a little bit deficient, and I do hit my head a lot. It could be something there. Now, if you actually look at the representation of the lips, this was a little bit ticklish, but you can actually see that the representation of the lips versus the face, that's the face, but that's the lips. You can see the lips are not my lips. You can see that the lips are greatly reduced. So it's much more close to veridical space of the face, so compared to S1. So the lip representation in S1 is very big. Rui-Song made a fine full bodysuit. This is a 64-channel air puff donnable bodysuit. including the face, yeah. And with that, we discovered that there were maps of the lower part of the body in LIP. So it actually goes all the way down to the toes. You notice the toes there, they're barefoot. So there's actually a representation overlapping the lower field with the entire bottom of the body, which is, if you normally can see your feet as you're walking, and if you block a view of your feet when you're walking over uneven ground, you'll feel a little uncomfortable, even though you're not looking down directly at your feet. If you look at the little homunculus, what you see is that you got this strange armless homunculus. It's got sort of a face and shoulders and chest and lower torso down to the toes, but the hand is kind of separated off in anterior interparietal sulcus, separated off from this kind of armless body. Now, we pretty much knew what we were looking for here because there was work in animals. If you look, this is a bush baby. If you stimulate that part of medial parietal cortex in a bush baby, you get lower limb movements. And there's another long series of experiments done with reaching in the magnet. The middle row is reaching in pitch black. So there's a very short visual target that goes off, and then it's complete pitch black. You make a movement toward a target in pitch black. If you look at the response during that movement to the target, massive response in parietal cortex and posterior cingulate cortex in what nominally you would think of as visual areas. So these areas are very much like bush babies. You stimulate different parts of parietal cortex, you get all different kinds of movements. We went on to map the inside of the mouth. There I am in the magnet with an SSFP scan saying pa 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 pa. And there I am saying cha 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 cha. It's a little disturbing. You can't see my teeth, but they're still there. So we'll add the teeth back in. So if you now look at a series of articulations of different positions in the mouth, so pa, fa, fa, ta, cha, cha, ka, you can see going back that way. What you can do is you can give people an auditory cue and have them articulate things going from the front to the back of the mouth. And if you do that, you look at the auditory cue, you can see the auditory cue, because that's happening like 56 times a scan. But the front to back's happening 8 times the scan. So that's what it looks like. So not expectedly, there's a map of the tongue and the palate in S1, but look in M1, there's a map in M1 as well. So even though M1 is controlling all these muscles in all different parts of the tongue, it's arranged as a straightforward front to back map in M1. And we did this properly in London with linguists saying very proper phonemes, and got the same result. And you can start to see the hint of other little maps, but the problem is you can hear yourself even over the noise of the scanner. And so there's always a problem of some tiny bit of auditory feedback. And we did put in white noise to try to cover it up, but still, even so, there's a little bit of bone conduction. So we did a lot of tonotopy. The auditory maps are quite different. They tend to be point-to-plane type projections rather than point-to-line type projections like you have in the visual system, because the receptor sheet is just one-dimensional instead of two-dimensional, like the somatosensory and visual systems. And so we did this early on, but got scooped by DiMartino, sadly, because we didn't publish my 1 year. But there you can see also some hints up there, like, what's that? Okay, so what we did in London was to use something similar to those windowed videos, which is bandpass filtered human vocalizations. These were emotional vocalizations, like laughter and crying and things like that, and bandpass filtered them because they're more interesting than just simple tones or white noise. Pop music, ABBA works great, completely flat spectrum. The record producers work it out, like if you're going to have some vocals, cut a little bit out for the vocals, put the vocal in there, just keep it maximum volume all the time. So pop music is actually very good for that purpose as well. If you look at the maps you get, they're quite extensive, and people have really overestimated the size of primary auditory cortex. We can now get a pretty good idea of where it is from our quantitative T1. It's very small, so these maps extend out a long distance. But you can also do the attentionotopy type maps. So there's in the left column, that's an actual tonotopic map, but the middle one is exactly the same spectrum, but the subject, again, professional musicians who are quite good at this, is just tracking different frequencies within that flat spectrum. And you can see something that looks just like the actual maps. So these maps are really attentionotopy or attentionatonotopy. They're not, they really require active attention to a particular frequency or a particular location to appear. So the auditory system really plays around with maps a lot. So if you look at, in the owl, you have frequency going this way in the nucleus laminaris, but the other way is characteristic delay by having axons conduct spikes in opposite directions. And that's the nucleus laminaris there. But if you go a little bit forward, you can see that the owl rotates its point-to-plane projections to generate a two-dimensional space map. So the brain is capable of sort of messing around with these maps. And we know in bats and owls that you have two-dimensional bi-frequency maps for the Doppler shift compensation behavior. There's been a lot of debate about what's orthogonal to frequency, but the bottom line is we haven't really gotten there to really convincingly say what the other axis is. There's several proposals, but it's very likely that given what we know about what the brain does with auditory system in the brainstem, that there's going to be a lot of messing around with, a lot of creative messing around with maps. So I'm going to turn now quickly to quantitative MRI to try to do in vivo architectonics and see how that relates to maps. So first, we'll talk about quantitative T1. So one of the problems looking at your standard data is they're just numbers. There's like 400 or 550. And there's a lot of variation. One of the things that causes it in a typical anatomical scan is variation in transmit flip angle, which changes the contrast. So you get changes in brightness, and FreeSurfer has a lot of things for flattening the brightness, but that can actually remove these small real brightness differences. And so it's better to actually try to combine scans. There are several ways of doing this. The one that got quite, this is just a picture of the actual 25% variation in flip angle at 3T. It's quite substantial. So there's several ways of doing this. One that got popular with Van Essen and the HCP was to divide these T1 by T2, but that's kind of a little grody and messy. So there's MP2 range, which is you can collect two sets of case-based data after one inversion, do some computations, or you can do our even more complicated one, which I'll take you through, which is you start off with the flash signal equation. That's sort of signal as a function of flip angle. And then the mathematicians turned this into that. It's unbelievable. That thing right there is just the same as that. There's some Taylor expansions and blah, blah. But it looks, especially for small flip angles, it might as well be the same equation. Much easier to deal with because now it's a linear equation. And so if you collect two different flip angles, you can solve for the T1 and the proton density. And that requires that you know the flip angle. So you do yet another scan. This is a 3D steam scan. So it's a spin echo and a stimulated echo, two different kinds of echoes. They have different dependencies. And so you can actually solve, given those two signals, for the local flip angle. It takes maybe about a minute or two. It's a relatively low spatial resolution 3D scan. And if you look at the data that you get out of that, what's the main advantage? Still alive. we have essentially found what Flechsig already knew to be the case in 1920, but the advantage is that the person is alive, and you can also more easily inflate the brain. And so I'll just go through some of the relations between this quantitative architectonics and the maps. So we can just begin to see sort of this typical pattern that you see in the cortex where it's dark and then there's a little bit of a transition and the upper layers are light. So here's an example of paging through the cortex from the white matter up to the PL surface. So this is near the white matter, and you can see in M1 and S1 there's a lot of myelination in the deeper layers of the cortex, but as you page up, you can see as you get toward the surface, there's a lot more myelination just in the upper layers in M1, which we know to be the case from invasive studies. So we can start to actually pick up some of these laminar differences between different areas. One of the problems with this method is that if you look at different areas in this direction, unfortunately the differences, the biggest differences you can get between different areas is unfortunately a lot smaller than the differences between the same area, different layers. So you really have to nail the surface sampling exactly in order to make this work correctly. Another thing is that if you look at gyri versus sulci, you can see that the areas kind of thin out in a sulcus and they sort of plump up in a gyrus. We didn't actually directly model that, we just regressed it out because it's correlated with curvature. And so if you regress it out, then you get a nice constant map across the cortex. And the very first thing we went to, of course, was MT, because that's the best area after V1. That little spot there in the lateral occipital is only about a third to a half MT. It's not all MT. So it looks round. We thought, like, MT is round. But yeah, it's only about half MT. So you get ipsilateral responses, which MT doesn't have in the front half of that. And you can actually see the retinotopy is reversing right in the middle of that little spot. We kind of knew that that was going to be the case from the monkey data. So you can see V6. Now, this was an area originally recognized by Flexig. He didn't name it, but it was originally recognized by Flexig. John Ullman discovered it had a big representation of the periphery in it, in owl monkeys. And slightly differently positioned, it sort of gets flushed dorsally by the V1 sort of flowing into the calcarine compared to a monkey. And it sort of pushes it a little more dorsally. It was a little hard to guess where it was going to be. But many years ago, Anders and I made this stimulus inspired by the late Ralph Siegel. And we found this area. It's just a giant response. We had nothing to, we said, like, we weren't expecting that. What is that area? So we put it aside for a while, but we eventually got back to it and showed that you can actually see there's a hemifield map there corresponding to that little area there. We didn't map the eccentricity axis until very recently. This required me going in the magnet, head off to the side, plastic screen about 2 inches from my face, 2 hours. But if you do that, you can see it's got a beautiful eccentricity representation that more or less starts at the point when people normally leave off their retinotopic mapping, because nobody would be so insane as to do what I just described. So if you look at you can see V6 right there has a nice big myelin signal. And then if you look right in front of V6, there's another area involved in reaching again, sort of visually driven reaching. You can see the VIPs and V3A quite easily with this method. And you can see differences at individual level. See, I have to turn the threshold up a little bit to make my MT visible, but I do still have one, not a very good one. You can see these huge differences between people. They're central tendencies, but substantial differences, and that we can actually pick them up now in a single subject scan. You can use it for doing better surfaces. So this is that really good data running through the regular free surface stream. It actually looks better if I just take that really good data, upsample it a little bit, down to like about 0.5 millimeter voxels, and just do a hard threshold. I get a better surface. So this is something I've learned interacting with MR physicists. Always better to have better data to start with than to try to fix not as good data later. Yeah, so you can use this. The interest in this techniques would be able to use them sort of for longitudinal studies or directly comparing data across centers, where you actually have a real T1 number instead of just 400. So you might notice that we didn't cover that many areas. And so one of the things I've been working on recently is to try to use gray matter diffusion. So if you look at the original architectonic papers, they're looking for these texture differences between different areas, like layer 4B, and different amounts of tangential and radial and even diagonal fiber structure in different areas. So you're trying to sort of characterize the texture, and diffusion imaging is good for that. A lot of people don't really pay much attention to it in the gray matter because they just assume that it's isotropic, but it's actually not isotropic. It's reliably slightly anisotropic. And so this might be a way to try to characterize how these areas differ at the level of sort of the local myelin structure. And you can do what many people do, sort of fit the diffusion surface with spherical harmonics and then try to make features, some of which are actually relative to the local surface normal. So you could sort of detect whether an area has kind of more tangential than radial diffusion or vice versa. This is way too many features. Some of these are rotationally invariant, but it's way too many features. We just want to make sure that we covered all our bases. And if you take a couple areas like that, you can recognize them in a second scan. So if you train on one scan, you can recognize them very, very well in a second scan. But you can also recognize never seen areas. So train on MT plus, distinguish that this is not MT plus, having never seen this data. But the other thing we started, that's the supervised method. You can also use unsupervised method. This is just k-means clustering of these diffusion features with no other constraints, no local area constraints or anything. And again, you can see test-retest reliability of this clustering. And so we took the HCP data. This was done by Tara Ganepola in London, and then redid their T1 divided by T2 analysis to make sure we were doing things right. Not anywhere near as good as the 2-flip angle. But if you look at that data and now look at k-means clustered diffusion feature data, you can see there's more stuff going on in it. The question is, is it good? But we can certainly recognize some of the basic areas like M1 and S1 very clearly, but you can see more distinctions in different little patches that might be just the beginnings of being able to do original cytoarchitecture, myeloarchitecture in living humans. And this certainly won't be hurt by higher field scanners, which are on the horizon. But what about cognition? So let's talk about cognition. So many years ago, with Don Hagler, we looked at this dorsolateral prefrontal cortex, where it's especially activated when you're ignoring location and doing a two-back task for identity. It's actually more strongly activated than when you're paying attention to location. But we had to map it. So we did this fine stimulus here where you face two-back while fixating the center. It's a really irritating stimulus. But and if you use this, you can actually see that you have a reliable map that persists over months. This is one subject scanned over several months. And in Ray Song's 24-subject average, you can see that there's actually two maps there in dorsolateral prefrontal cortex, and probably 2 or 3 in the frontal eye fields there. So those are constant enough that they show up across 24 subjects. So that again addresses the problem of there's some variability, but there's enough constancy so that you can actually see something. It doesn't average into a mush. How does this relate to cognition? This is an experiment where we had people imagine walking around the Coxeye building, or imagine stopping in certain places of the Coxeye building. If you look at the red activated areas when you're imagining navigating, and look at those outlined in yellow, you can see that those things overlap a lot of these maps. So just imagining moving around in the Coxeye building activates neurons that are predominantly in areas that have these topological maps. But that's just navigating around. What about language? How does language overlap? So here's parafoveal reading versus fixation, and you can see the usual suspects. There's like the dreaded visual word form area in infratemporal cortex, and then there's Marty reading, where you fixate the center and slowly have the story about Paul Klee go around the center. If you look at that, you can see the visual word form area is retinotopic, as it should be. If you map with this kind of stimulus, it's got numbers and letters in it, it actually doesn't activate the visual word form area. So it requires actual words at different locations in order to activate it, which you knew from block design studies. But it still has a degree of retinotopy. And again, why shouldn't it? Because you got to look, you're saccading around. If you read scientific papers the way I do, it's like dun, 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 dun. You got to go to all different places, to check out where that word is. Okay, but what about comprehension, language comprehension? That was just like reading. So here we have to get all maps. So we do some visual maps, some auditory maps, some somatomotor maps. We have people actually gently move different parts of their body. So here's all the maps in a single subject. There's the visual maps, both hemispheres flattened. There's sort of more or less where some of the main ones are. There's the central sulcus. Look for the central sulcus and the STS to sort of orient yourself. Here's a body map. So you can actually see the VIPs appearing there. You can see a little bit of AIP appearing. You can see the S2s showing up. You can see the supplementary motor areas showing up. There's a mouth of the map in this subject. A map of the mouth in this subject. There's the lips going down to the throat. There's auditory maps in this subject, and so we'll just paste them all together. So there's the auditory maps, the visual maps, plus the somatomotor maps, plus the mouth map, plus the auditory maps. So we have all our maps. And then we can do a controlled reading. So it's rarely been the case that people actually do the kind of reading that people really do when they're reading, like you move your eyes. So here we just had people read these word length controlled fixations of little paragraphs versus reading Hindi transliterations of them, which don't make any sense, but the saccades are exactly the same, and the word length is exactly the same, and the fixation duration is exactly the same compared to a dot. And so then you can get sort of English versus fixation, so comprehension versus absolutely just fixating, but you can also do English versus doing all the kinds of attentional things you would do, but actually comprehending something. And then we can compare that to all maps. And again, you can see a substantial amount of overlap. So here we'll give the reading the benefit of the doubt, sort of just reading versus fixation. And you still see there's a substantial amount of overlap there. And so we did this with 20 subjects, the same subjects. So we took the same subjects, did all the visual maps. That's the amplitude of them. There's all the retinotopy. You could actually pick up finally the dorsomedial-frontal eye fields. There's a map in the dorsomedial-frontal eye fields that we were able to pick up. This is multiband data, so we were able to go 4 times faster because of the good data that the physicists thought up, little tricks of trying to collect data more rapidly. Here's the tonotopy average. Notice there's the dorsomedial-frontal ear fields. I just named those. So they're attached to the supplementary motor cortex. They have an auditory map in them, and they're actually activated. You'll see they're activated during reading. So if you look at the visual and frontal cortex maps versus the auditory ones, you can see that they're largely non-overlapping, even in the frontal cortex. Here's the somatomotor maps. They're a little noisy because the person is moving their mouth, and so there's a little bit of mouth motion artifact here and a little bit of self-auditory stimulation here, which slightly messes them up. But if we just sort of gray those out, and then look at where the dorsomedial frontal eye fields are and the dorsomedial frontal ear fields are, you can see that they're basically surrounding these 2 supplementary motor areas in middle cingulate cortex. And then we have them do this again, all the same subjects. And so then what do you get when you do that reading experiment on average on the surface? The usual suspects, like some parietal cortex area and dorsolateral prefrontal cortex area and STS and infratemporal cortex and dorsomedial frontal ear fields when you're reading. And then you can actually do some overlap. It depends on your threshold, of course. We're doing sort of retinotopic mapping in one case versus block design in the other case. The little difficult to directly compare them. But at various different thresholds, you get a reasonable amount of overlap. And so the bottom line is that something like on the order of 80% of the activation during comprehending versus not comprehending overlaps maps of some kind. So we also did the very same thing with picture stories. So if you look at picture stories like this, where you're being directed around by a Gaussian sort of bubble-like mask to read the story versus being directed around a scrambled version or just a temporally scrambled version, you can look at how wordless serial visual comprehension overlaps with maps. And it looks more or less like language comprehension for the most part, less stuff in the SDS. So there's the language plus 3 maps. Here's an overlay of the scene comprehension versus moving your eyes and attending to things and doing everything else except not comprehending anything because it's all scrambled up. And you can see that there's a lot of overlap between the scenes and the language, and there's a lot of overlap between the scenes and the language and all the maps. So yes, there's a lot of overlap there between these maps and real cognition. Now, in the last 10 minutes, we're just going to race through the first pictures of the cerebellum. So 75% of the neurons in your brain are there. So why haven't people dealt with it? Well, it's small, and the other problem is that if you look at the cortex, the neocortex, it's relatively thick. Well, you're not really resolving the cortex in the cerebellum. This is just one of the main lobules, but you're not actually seeing the cortex. So we got a dead one and scanned it at 9.4 T for several nights. So that's what it looks like when it's alive. That's me there. This is what it really looks like. That's a scan of a dead cerebellum. So this is a T2 star-weighted scan and a proton density scan. And then I combined them and added extra sauce. See how much better that is after the extra sauce. And then that was good enough to actually be able to reconstruct it. So this is just going through this insane structure. It's a giant structure. 75% of your neurons are there. Here it is going from the top down. And this was the 512 cubed scan. And here it is going from left to right. You can see that's one of the hemispheres of the cerebellum. You just went through the dentate, and then that's the vermis right there, and now you're in the other hemisphere. Just an insane structure. So it was a giant surface, 5 million vertices, 25 times as many vertices as we need for like a regular puny neocortical hemisphere. You actually need them because this is the scale you're talking about. That's the actual 2-scale size of a Purkinje cell, which is perpendicular to these folia. So I tried it the other way, didn't look as good. So if we take that cerebellum and inflate it, you can see it doesn't inflate very well because of the way it grows. There's all these little bubbles in the cerebellum. It's actually quite difficult to inflate this thing. Those little stripes are all the little folia so the folia inflate, but the main lobules don't inflate. And so what are we going to do with this? So this was so we took out the free surfer chainsaw, sort of cut through it a bunch of places, and then cut it into a couple pieces. So this is the anterior lobe. This is what it looks like when you cut it so you can flatten it. There's the primary fissure at that white arrow, which we're just passing now, and then going into the posterior lobe of the cerebellum. And then you go into the, further in the posterior lobe. This is Crus 1 and Crus 2, sort of the cognitive cerebellum. And then you end up down in lobule 8B, and then finally lobule 9, which is the cerebellar tonsils. So it's over 1 meter long. So it's 6 centimeters wide, and you just stretch it. It's just a crinkled thing. You can stretch it out. It's almost almost 5 feet long once you unfold the thing. So what's in there? Well, you've got to do even finer grain maps than I was doing in the cortex to pick up the structure. So there's all these little patches in there. There'll be like a little retinotopic, somatotopic patch of some little patch of the mouth next to a map of the paws next to a little representation of some part of the back. So there's these little fractured somatotopy maps. So what is the scale of these? Well, here's the human cerebellum. They're, like about that big. So, so more work is required. But the important thing to take home message here is that the human cerebellum is expanded just as much as the cortex, even slightly more relative to a macaque monkey than the cortex. So it's just as much of our humanity as the neocortex. It's just as much of sort of characterizing who we are. Almost, very, very much less studied than other parts of the brain. Okay, so in the last 5 minutes, let's talk about this experiment. So Esther Kuhn came to me to do an experiment in Leipzig, collaboration with the 7T scanner at Leipzig. I didn't think this would work. Of course, it involved maps, and so I liked it. I wanted it to work, but I didn't think it would work. And so that counts as the best kind of data where you really didn't think it was going to work out, but it did anyway. And so I'll tell you what this experiment is. So this is mirror neurons gone wild. So what it is, it's a very simple experiment. Actually have somebody touch your hand with sandpaper versus look at somebody touching somebody else's hand with sandpaper, or in third-person view, or almost touch their hand with sandpaper. The trick was to do this in a periodic fashion so that you could actually get a map of the actual touch and perhaps a map of visually induced empathetic touch. So we'll just compare these two here, just the actual touch to the first-person view of touch. So we had to improve our pipeline here. So these are 7T data, 1.5 millimeter squared voxels, all female participants. And so we're doing circular correlation, vertex-wise circular correlation on geodesically defined nearest neighbor searchlights, taking two sets of complex valued data, and then only using representative vertices so we don't double dip. And so that gives me a chance to show some colored pictures. So these are the voxels. These are voxels intersected with the surface. But if you actually look at the surface mesh, it's finer. And so it's kind of cheating to calculate cross correlations if all these samples came from the same voxel. So these are the representative vertices. So those are the vertices nearest the center of the vertices that sampled one functional voxel. And so that's a fair cross correlation. Here's the searchlights now. So searchlights sort of moving around. And if you do that, you can really see there's a strong little spot of map correlation right in S1. And you can actually see this is the actual visually induced map in S1 and the actual touch map, which is a much larger amplitude response in S1. So if you sort of zoom in a little bit and turn the threshold down, you can see there really is, there's almost nothing else across the brain that actually shows a correlation between the visual map and the somatosensory actual touch map. So it's present in area 3B and also, not just area 1, but also 3B, so the primary input area. It's not a big signal, as it shouldn't be a big signal, because otherwise every time you saw somebody else get touched, you would actually feel a direct touch. Could be an inhibitory signal for all we know. But it's probably about 10% the size of the actual touch signal, but it's a good signal, and it slightly varies as you look at different layers. You can see that there's a bigger laminar variation in the actual touch signal compared to the visually induced signal. There's a lot of, it's in its 10th review, which means it's really good. It's really true. That's why it was so hard to get through. But one of these days, there's been appeals. Eventually this will Eventually this will get out. But we did EMG to make sure that the person wasn't moving at all during this. The almost touch doesn't work. That was very, very nice. Third person works pretty well, despite the flip. So the visual stimulus is suddenly rotated 180 degrees. So the retinotopic stimulus was flipped by 180 degrees, but it still works. We can't tell excitation inhibition, and we need smaller voxels. But in conclusion, the medieval cognitive neuroscience guys were wrong. So what is this theory? This is the original cognitive science theory. You've got all these modalities. They come together in the common sense. There's no maps at all. And you have all this sort of like, warp drive that's doing all the cognition. This is where math is, and then memory's at the back. It doesn't look like that. There's maps all over the place. If you've got a map, it doesn't mean that it's not good for cognition. And that's often kind of the implicit idea you get from reading cognitive neuroscience papers. And just think about visual experience. It's sharp. It looks like V1. It doesn't look like IT. It doesn't look like a giant receptive field smear of everything. You look out, you see fine details, eyes, little, close your eyes. Try to imagine that. It's not anywhere near as good. So cognition probably does, penetrate all the way back to these early maps. It's involved in these early maps. Don't throw them out. They're good. They're worthwhile. So I'll just end with a picture of our magnet being delivered. So if you want to come and play around with a Siemens Prisma, a nice Siemens Prisma, come and check us out at SDSU. So there's lots of people that did most of the work that I'm talking about here. And too many to mention, and I'll just leave you with one last picture of where you get some kind of retinotopic visual modulation in the brain. So you can see, there's maps all over. Thank you very much. Terry.
What fraction doesn't have maps?
Wait, the dark
Oh, sorry.
so you gotta use the mic.
Right. So it's wonderful to see all of the maps and that they're activated with cognition. Roughly what fraction of the surface area doesn't have maps?
Doesn't have maps. So it's on the order of a third that doesn't have straightforward sensory surface maps. But of course, we know from something like owls that you've got the space map, a constructed space map. That's not related in any straightforward way to the sensory surface, and so it could be the case that there's maps there. We just don't know know how to parameterize them.
Let's take that case. What has anyone looked at the auditory spatial map? That from auditory localization, has that been mapped out in the cortex?
They've looked for that a little bit, just trying to see if they had sort of inhibitory excitatory columns kind of thing, but no great successes. There was a few studies where they tried to put stimuli behind the head because you actually have a pretty good map of whether things are coming from behind your head or in front of the head. So there's one study that suggested that you actually could distinguish behind the head from in front of the head in the brain data, but yeah.
Do you think that there might be maps having to do with rewards, so that, say, in the nucleus accumbens, you might find, or somewhere in the caudate, you might find maps of high value, middle value, low value, avoid at all costs?
Well, that's sort of the general question is, what about other? So I would break that into sort of three categories of maps. So we have like straightforward, simple to find maps that are based on sort of attention to receptor surface position. And then we have various kinds of constructed maps that are something like, say, the space map or something like that, like a reward map or some kind of higher level construct that you've actually played out across a map. And then there's the possibility, of course, that maybe there isn't a map, maybe there's actual spatial structures, little propagating sort of, glider type patterns that are map-like but are not attached to a particular portion of the map. In terms of sort of the layout, we account for about like 2/3 of the cortex with reasonably straightforward sensory maps, and certainly there's a variety of different higher level maps that nobody's been smart enough to sort of, discover yet. I mean, another example. So I'm not answering your question at all, but we do know at least the maps in the dopaminergic part of the substantia nigra are related probably to different motor structures. So those are kind of more like motor sensory maps as opposed to the kind of higher level ones that you were asking. But those things could definitely be there. You've got the hippocampus, you got the entorhinal cortex. There's a lot of other tricky areas. But I'll just say one thing about this one study that I was talking earlier about, which was this optical recording from Tanaka's lab of the face pose. And it hasn't been duplicated for other objects, but what they found was that if you actually if you take a plastic mannequin head and slowly rotate it in front of the monkey, you see these little spots that sort of just translate on the infratemporal cortex. And they tried it with other objects, didn't really work. And so there's a map of face pose. You move the face around, nothing happens. You rotate it, and you see the little translation of the spot. There should be more of those. I mean, if you think, I'm just sitting here, I'm providing like high quality OpenGL gamer input at 1,000 frames per second, and I've got this giant training set of the world, of how, and I've got my camera perspective in there too. I'm moving my camera around, and it rotates and translates and does very predictable things. Why do we have more sort of OpenGL in the brain? Because you've got this giant training set that would be very useful to know like what this looks like in a different pose. So that's one example I can point to, but that hasn't really been generalized.
Has anybody looked at mental rotation?
They have, yeah. There's several things on mental rotation. Not real mappy.
Marty, can you speculate as to how the grid pattern maps in entorhinal cortex might interface
with these? That was another question that I just got in the earlier discussion. So obviously, if you take a very simple, say a simple head direction cell model like Ketchen's original 96 model. So you've got a ring of cells, and given junk, they make a little bump. And so it doesn't matter, where, there's always going to be, I think the direction is such and such, even though I have no idea what the direction is. Now, if you just take that ring and put crap into it, just junk into it, just like, well, yeah, it's some little visual thing slightly over on the right side, or some green thing, or some pokey thing over here. You put just like real low quality sort of junk into the ring, you might be able to then, get a relatively high resolution response out of the head direction cell once you add in, a bunch of, vestibular input. So the vestibular input is nice and round, and it's kind of pushing the bump around. And then you've got these cues. You've got these sort of not very good, instead of crap, visual cues sort of coming into different parts of the ring. And so it could be the case that a map like that doesn't require super good retinotopy. In fact, just any old retinotopy is probably good enough to essentially fix different parts, fix directions to different parts of the ring. And of course, you could imagine the same thing in the 2D case for place cells or for grid cells. So how that all works, I mean, that's a tall order to try to actually. What you really want, and all you want, is just like all of the cells and 2 or 3 synapses away that came into this one grid cell to like find out how it got that way. But yeah, it's a tough order to imagine 2 or 3 levels of retinotopic areas, before you actually get to the entorhinal cortex.
Marty, I have a question about how these are established. I mean, it seems like there must be layers of maps coming in at different times during development, and they're interacting in some way so that they're related to each other as well as being maps in their own, on their own. Have you looked at how the mapping develops in time,
So
in
we've done
monkeys
some
or
of that
in-
relatively recently. Just the problem is we haven't looked early enough. So we've gone down to about 6 years old. And so if you go down to 6 years old and look at retinotopic maps and population receptive field maps, as far as we can tell, they look completely adult by 6. So we found other things that don't look completely adult. So regarding like language or object perception, you can see some differences. But as far as the maps go, we haven't seen anything in the posterior maps. Now, it could be that we didn't do the frontal maps in that case for various reasons. And so it could be that some of the frontal maps develop a little bit longer. But there's often sort of a knee-jerk reaction that the V1 must be genetically or somehow set up by gradients, but frontal cortex couldn't. But why not? You know, I mean, I think it's very reasonable to me that dorsolateral prefrontal cortex has some kind of predisposition to form a map. If we can actually see it across 24 people, it's probably a pretty robust sort of mapping structure. But that is about to be done by more people to try to actually look, especially at some of these higher maps. I think Colin at Grill Spectre recently had some data suggesting that some of the face, not the maps, but at least some of the face cell responses in frontotemporal cortex in humans do show some development where you can see some difference. But it's been hard. I mean, for the most part, whenever we look, stuff looks pretty much like you'd expect it to look in an adult. You got to go quite early, like 2 or 3, and that's hard. You really need probably some kind of prospective motion video correction to be able to, you know, deal with the younger, really young kids. Marta.
So most of my questions all would deal with individual differences. So I'm curious, on the one hand, about the study you thought wouldn't work, where you taught the last one. Do you think that you could get an independent measure of empathy in that individual and see and in individuals, and then correlate that with the degree to which there's overlap between the two areas? I mean, the maps.
Absolutely, it's extremely strongly correlated with that. So there's huge variation in empathy. I mean, that was why only women were used in that study, because they generally tended to.
So the other question is, in general, do you think you could make some generalizations about, for example, to synesthesia or to how good an athlete somebody is going to be, or how good a musician they're going to be, as a function of the overlap of the different mappings?
That would be another interesting one to try. And there are enough variations, like almost everybody has a V3. I have an owl monkey-like V3A touching my V2. I don't have a V3. So there's probably, I don't know exactly how I got that way. I was a normal enough child. So there are some variations like that where there's actually major differences in the neighbor relations of maps, of just even early visual maps like V3. That's only 3, you know. So people they've started to do stuff with this, but a lot of it is, the quality of it wasn't so good. And in some of the most straightforward ones, like Jeff Boynton years ago looked at vernier acuity. You can see variations in that. And then he got somebody in his lab who had like a V1 that was half the size of Jeff's V1. And his vernier acuity was like a little bit worse, but not very much. So the things people have done so far, they haven't found as big effects as you might like in some cases. But I think it's just the start, because people, a lot of studies, they just grab another set of people and don't, you know, bring them back 20 times. And that's really the kind of study you need to do, like we did with the language study, where you just bring the person back and do the maps in detail. And then that already took like, you know, four whole scan sessions, and then do some other scan session on top of that. So it's you really have to sort of hit it hard with individual subjects to make these things work. And people are a little bit more hit and run, myself included.
It's for the people at Dartmouth. It's for the other people.
Did you do things? No, did you find or find something which is done by or certainly done by not good function? So do you have a normal ear on the left and a non-normal or something like that. In Tübingen, for instance, people look on different eyes, left versus right. Or, I mean, the less probably the more you can understand. Would you think so?
Are you asking whether or not there's like behavioral effects on the maps, or maybe behavioral effects as a result of the maps, or a result of
the result of the
result of changes in the maps?
Yeah.
So we haven't done very much with that. Probably the largest number of studies was just looking at deaf subjects. And so one example, I could just tell you one example with regard to deaf subjects. So when you're signing, you tend to look at the person's face, and then the person is signing in the periphery, but not the really far periphery. It's generally sort of parafoveal area. And so initially, to try to get a look at that, we did some mapping, visual mapping of deaf subjects, and looked at the estimated population receptive field size as a function of eccentricity in early visual areas in deaf subjects, deaf from birth subjects, and hearing signers. And what we found was, it's kind of exactly the opposite of what we found. So it's a really good piece of information. So what we found was that in the deaf subjects, the population receptive field sizes in the periphery were bigger. So we would have expected, since they're relying on sight to deal with stuff, cars flying from behind them, that they might be smaller or they might be finer grained. But then when we thought about it a little while, of course we came up with an explanation for it. To go back to the late Walter Heiligenberg, if you imagine a bunch of relatively large receptive fields, that will give you a more robust estimate of exactly where something is than narrower. So you actually want a little bit more overlap. And so these deaf subjects, in their far periphery, far outside of the periphery of sign, seem to have plastically changed V1 to have larger effective receptive field sizes, presumably because they're walking along and a car is like coming up like right in the absolute far periphery, and you really need to pay attention to that because you have no auditory signal of where it is. And so that was an example of where it looks like the modified periphery has resulted in a change in the map. I mean, the maps look normal, they just look like the receptive fields are a little bigger.
So for these maps you've been showing, they're primarily maps of some sensory input that's either two-dimensional or one-dimensional, which is sort of relatively straightforward to imagine how that's painted onto the brain. Presumably at some point the brain computes a representation of the world in 3D. And if that's the case, is there any thought about how once the representation gets to 3D, how that might be mapped, or if it's mapped physically onto the brain at all?
Well, that's a very long-standing assumption. And if you look at sort of the whole sort of history of computer vision, people assumed like there got to be some kind of three-dimensional OpenGL in the brain somewhere that actually that models objects and knows about sort of simple, linear transformations. It's been surprisingly hard to find at all. I mean, if you look at the higher level infratemporal cortex, which is kind of more or less the top of the system, like Nikos has looked at that, and what he found was you train monkeys on random looking little paperclip things, and initially you find there's not many neurons that like them, and after they've been trained on them, they start to like them, but they still are basically view-based, and so they never found sort of like the OpenGL model that actually had the three-dimensional, I
guess the backwards version of that question is it could be that that fact is related to the fact that 3D can't be mapped onto a 2D cortex?
No, I hadn't thought of that, but that's a possibility too.
So we know that disparity is mapped within the map as an extra feature.
Yeah.
So just because it's not on 2D surface doesn't mean it's already is not being represented there. It's just that we know that there's additional features, and that
Yeah.
might be the solution, is that's at a finer scale.
Yeah, on a finer scale. Yeah, no, I mean, definitely in many of these areas, you have an embedded orientation map, embedded disparity map, embedded direction map. So you definitely have these submaps in there. But the other thing about the 2D and 3D, if you look at the somatosensory cortex is, if you look at the shape of the body, it's a ridiculous thing. How do you get that onto the somatosensory cortex? Lots of cuts. You know, like if you actually look at the somatosensory cortex, it's got hundreds of cuts in it. It's like you snipped out the underside of the finger and pasted that down, and then you took the back of the finger and put this part, you know, over here and this part over here, and then the palm is over here. So you got like hundreds of discontinuities in the somatosensory system. Yeah, there's thousands of cuts in there. So the brain will find a way. There's the star-nosed mole that has a little star shape in its cortex, but you know, if that doesn't fit, we'll find a way to jam it in there. So I think, you know, when it comes to sort of three-dimensional objects, there's probably a way of putting it in there, but it's still surprising to me, given like the high-quality training set, that you don't have OpenGL. That nobody's found OpenGL in the brain. Maybe they just didn't find a way to you know, directly measure it yet, but yeah, it's kind of surprising. You'd assume that at some point there's got to be some kind of mapping, but you know, biological systems always take a shortcut whenever possible.
On one of your auditory tonotopic maps, I thought I saw a little spot down in the fusiform gyrus, and I wonder to what extent do you see auditory activation of the visual cortex, and what does it look like?
So that's a good question. So every once in a while, we pick up a tiny little bit of auditory in the fusiform gyrus. But I'll just say, when you actually look at the majority of the auditory maps, they go down into the STS. Go down to the STS, and they kind of stop near the STS. Sometimes get slightly onto the lower bank, but they usually don't get onto the fusiform. But there's often a little bit of a trace of something in there. So it's possible that there could be something in there. One thing to consider is when you actually look in fine detail, like Seltzer and Pandya have done these experiments where they injected both auditory and visual and looked at the STS. And surprisingly, what they found was the modalities were stubbornly immiscible. So you'd inject the sort of superior temporal gyrus, and you would see like a little, you come down into the STS and everything's still, we're all auditory here, everybody's good, you know, and then you run into a little patch of visual with no auditory, and then there's a little bit of auditory beyond it. And then you inject infratemporal, you see the same thing. You'll run into like, you know, a little auditory patch, don't connect with that guy, it's wrong modality, then jump over, and then there's a little visual patch in there. And recently, Rainer Goebel and those people they have a 9.4T human magnet. They've tried to look at the STS, and when they really home in on it, it's got these tiny little patches that seem to be somewhat modality specific there. But back to the fusiform, yes, every once in a while we see a little bit of auditory stuff down there, which is very, you know, redolent of ideas about all different kinds of, you know, synesthesia down there. It's not a big signal, and it doesn't show up in everybody. It doesn't show up in the average.
So we actually have some refreshments, and we can, that's fine, head outside and thank Marty very much for a great talk and nice discussion afterwards.
Thank you. Thank you for having me.
Yeah, so please join us.